We will start. Good evening, good afternoon, good morning, everyone. Thank you very much for joining us for this ISHR JMCC webinar. My name is Junjie Xiao from Shanghai University. I am the social media editor of JMCC. Today, we will introduce our editor's choice selection paper from the corresponding author, Professor Nie Yu. Professor Nie Yu is from Fuwai Hospital in Beijing. He has published many papers in journals such as Circulation, Advanced Science, Cell Reports. He has been awarded the Paul Dordella White International Scholar Award from AHA. His major research interest is cardiac regeneration. Today, his PhD student, Dr. Chenzhen Huang, will introduce their work. If you have questions, please leave a message. Let's welcome Dr. Chenzhen Huang to give the talk. Thank you very much.
Okay, thank you. It's okay. Hello, everyone. Good evening. Thank you very much for attending today's report. I'm Chenzhen Huang, student of Dr. Nie Yu. The title of our report is The Longevity Protein P66 is required for neonatal heart regeneration. And I'll report from the following 5 aspects. The self-renewal ability of adult cardiomyocytes is not enough to effectively compensate for the lost cardiomyocytes caused by myocardial infarction, eventually leading to heart failure. So, inducing endogenous cardiomyocyte proliferation to replenish the lost cardiomyocytes is considered to be the most effective approach to treat patients with heart failure. Unlike the adult mouse heart, the neonatal mouse heart has the ability to regenerate completely after injury, which provides an ideal model for studying the mechanism of heart regeneration and find therapeutic target for cardiac injury. Studies have shown that senescence signaling play an important role in neonatal heart regeneration. In neonatal mice subjected to apical resection, a mode for heart regeneration, senescence signaling was identified in the peri-resection regions after apical resection, but disappeared when the hearts were fully restored, and clearance of senescence signaling by genetic deletion of tumor protein 53, TRP53, impaired regeneration after apical resection, indicating that senescence signaling is responsible for neonatal heart regeneration. Therefore, a better understanding of the role of senescence signaling may help in the identification of novel targets for heart regeneration. The longevity protein P66SHC, together with P52 and P46, belong to SHCA adapted protein family. Among the 3 isoforms, P66 is the only protein that plays a critical role in senescence signaling by causing alternation in calcium response and 3-dimensional structure of mitochondria. In addition, P66 also participates in multiple biological processes, including reactive oxygen species production, cell proliferation, and cell aggregation, and cell adhesion. However, as a critical determinant of senescence signaling, the role of P66 in heart regeneration remains to be elucidated. So, we want to know what role does the longevity protein P66 play in cardiomyocyte proliferation and heart regeneration. and to investigate whether P66 regulates heart regeneration. Neonatal P66 knockout mice and their control wild-type litmates were performed apical resection on postnatal day 1 and sacrificed to collect hearts at 7 days post-resection and 21 days post-resection. Medicine staining showed that heart regenerative capacity was suppressed in P66 knockout mice relative to wild-type litmates at 21 days post-resection. Compared with the wild-type group, the heart function in P66 mice was significantly decreased at 21 days post-resection. As we all know, heart regeneration is based on the activation of cardiomyocyte proliferation. To investigate whether P66 regulates heart regeneration through cardiomyocyte proliferation, we co-stained the cardiomyocyte marker alpha-actinin with pH3 Ki67 marks of cell proliferation respectively. We found that cardiomyocyte proliferation was blocked in P66 knockout hearts at 7 days post-resection. We first confirmed the effect of P66 on cardiomyocyte proliferation. Primary neonatal mouse cardiomyocytes were transfected with siRNA to knock down P66. And we confirmed that P66 was knocked down. EDU incorporation assays showed that P66 knockdown suppressed DNA synthesis in cardiomyocytes. And we confirmed the decrease in cardiomyocyte proliferation by staining alpha-actinin with pH3 and Ki77 respectively, without apoptosis. Enhanced glycolysis is beneficial for cardiomyocyte proliferation, and our sequence data revealed that P66 knockdown decreased glycolysis and increased oxidative phosphorylation in cardiomyocytes. Our results indicated that P66 deletion causes cardiomyocyte cell cycle arrest and glycolysis inhibition. And so far, we know that knockout of P66 resulted in incomplete heart regeneration after apical resection in neonatal mice. And knockout of P66 blocked cardiomyocyte proliferation after apical resection in neonatal mice. Also, knockdown of P66 blocked cardiomyocyte proliferation without apoptosis in vitro. So next, we want to know the mechanism of P66 in regulating cardiomyocyte proliferation. To explore the examined mechanism, we analyzed the gene expression profile by RNA-seq. RNA-seq analysis showed that 620 genes was upregulated and 829 genes were downregulated in the P66 knockdown cardiomyocyte group compared to the control group. And geoanalysis revealed that ARCs and canonical wind signaling pathways were changed after P66 knockdown. And for further validation, we performed Western blotting analysis of total beta-catenin. And beta-catenin is the downstream effector in the canonical wind signaling pathway. And we confirmed that only the wind beta-catenin pathway was significantly downregulated in response to P66 knockdown. And importantly, P66 knockdown suppressed the phosphorylation of beta-catenin on serine 675, which promoted beta-catenin transcriptional activity. And to further confirm whether beta-catenin is a functional target of P66, we knock down the beta-catenin gene CTNNB1 in primary cardiomyocytes by siRNA immuno.
I think there are some problems with the internet. Hi, Dr. Huang. I think there are some problems with your internet.
Hey, Professor Xiao, let me contact contact Huang. Okay.
Thank
Moment,
you.
wait a moment.
Thank you. So let's wait for a while so that Professor Nieyu contact Dr. Huang for the connection of internet.
Why? My Wi-Fi is.不是，把你的耳机借我一下。OK，等一下。It's okay now.
Okay, maybe start. Yeah, maybe
Oh.
start from page 17, I think.
17. Here.
Yeah, yeah.
Oh, okay. Sorry. Okay. And we want to confirm whether beta-catenin is a functional target of P66. So we knock down the beta-catenin gene CTNNB1 in primary cardiomyocytes by siRNA. Immunostaining showed that beta-catenin knockdown suppressed cardiomyocyte proliferation, and this is consistent with the effect of P66 deficiency. In addition, overexpression of active beta-catenin attenuated the suppression of cardiomyocyte proliferation by P66 knockdown. Our results demonstrated that P66 regulates cardiomyocyte proliferation by activation beta-catenin. When the beta-catenin signaling has been shown to play an essential role in cardiomyocyte proliferation. And so, deciphering the mechanism by which P66 influence beta-catenin expression and activation might provide important insight into cardiomyocyte proliferation. Because when the beta-catenin signaling regulates the expression of cyclin D1 and c-MYC, we detected the expression of these proteins in P66 knockdown cardiomyocytes. Our Western plotting showed that only cyclin D1 is significantly decreased, and no warp wires changing was observed in c-MYC. And we then overexpressed active beta-catenin in P66 knockdown cardiomyocytes, and we found that cyclin D1 was upregulated. This suggested that cyclin D1 might be involved in cardiomyocyte proliferation regulated by P66 beta-catenin module. And so far, we know that P66 knockdown by siRNA regulated gene expression profile in primary neonatal mouse cardiomyocytes. And P66 regulates cardiomyocyte proliferation by beta-catenin cyclin D1 module. In summary, we found that P66 deficiency decreased neonatal mouse cardiomyocyte proliferation and impeded neonatal heart regeneration after apical resection injury. And RNA-seq and functional verification demonstrated that P66 regulated cardiomyocyte proliferation by activating beta-catenin signaling. And our work broadened the understanding of the role of P66 and suggested that P66 performs essential functions in cardiomyocyte proliferation and neonatal heart regeneration. These findings also provide new insights into senescence signaling in heart regeneration. And that's all. Thank you all for your listening.
Thank you. Do you want to do the acknowledgements? Okay. Thank you very much for the nice introduction. And I think we can start the questions and answer stage. So if any audience has questions, please put your questions. I think in the bottom of your screen, somewhere you should have a place for questions and answer. Maybe we wait for 3 minutes for the questions.
Okay.
Okay, we get one question.
Okay, thank you. For the first question, P66 may affect heart regeneration, also regulating non-cardiomyocyte cell types such as fibroblasts. Thank you your question. And we also confirmed the effect of P66 on the non-cardiomyocyte cell types, but we found that P66 had no effect on non-cardiomyocytes such as fibroblasts or other cell types. And thank you. So, the second question, how does P66 regulate cardiomyocyte glycolysis? Through our experiment, we think maybe P66 regulate cardiomyocyte glycolysis through HEF1 or some other signaling, such as oxygen species production, maybe. But we may conduct more experiment to confirm this the mechanism, how that P66 regulate cardiomyocyte glycolysis. From our data, we know that P66 regulates beta-catenin, but how P66 regulates beta-catenin need more experiment. From some studies has shown that P66 regulates beta-catenin through oxygen species, maybe in cardiomyocytes. P66 regulates beta-catenin through oxygen species, but I need to conduct more experiments to confirm it. And in adult myocytes, we don't confirm whether P66 regulates beta-catenin in cardiomyocytes and in adult myocytes. Thank you. How did you find pieces to regulate?
We missed the acknowledgement page. Chenzhen, hello. Sorry for that. We missed the acknowledgement page, and this work is we finished together with Professor Hou Zaochen, and this paper was supported by some grant, such as let me see. National Key Research and Development Project of China, and this should be show it in the acknowledgement page. Okay, let me explain it. Sorry for we missed this page. Okay. Chen Zhen, continue.
Okay. The next question: how did we found PC6 regulate cardiomyocyte proliferation? And in our virtual experiment, we found that P66 is important for heart regeneration, and because we all know cardiomyocyte activation is important for heart regeneration. So we examined the effect of P66 on cardiomyocyte proliferation, and we found that P66 knockout suppressed cardiomyocyte proliferation significantly. So for further confirm the effect of P66 on cardiomyocyte proliferation, and so we isolated cardiomyocytes from neonatal mice and knocked down P66. And then we examined the effect of P66 on cardiomyocyte proliferation in vitro through EDU incorporation or cosine pH3 and Ki67 with alpha-actinin. We confirmed that P66 knockdown decreased cardiomyocyte proliferation in vitro. So we know that P66 regulate cardiomyocyte proliferation in vivo and in vitro. So we confirmed that P66 regulated cardiomyocyte proliferation. Thank you. Angiogenesis. Yes, P66 may affect heart regeneration through regulating angiogenesis. But in the future, we may confirm that whether P66 affect heart regeneration through angiogenesis. Thank you. Okay, from our unsick result, we know that the 621 upregulated genes are focused on canonical wind signaling pathway, and also X signaling pathway is regulated. But our Western blotting data showed that P66 regulated cardiomyocyte proliferation by wind signaling because the beta-catenin is effector of wind signaling, and P66 knockdown in cardiomyocyte downregulated beta-catenin and also downregulated beta-catenin phosphorylation. But X signaling is changed without significantly changed. So we confirm maybe P66 regulated cardiomyocyte proliferation by wind beta-catenin signalings, and also we confirm the effect by overexpression active beta-catenin. Because overexpression active beta-catenin can upregulated the cardiomyocyte proliferation, which suppressed by P66 knockdown. And thank you. How do you think PC is related to the exact? Thank you. Through our experiment, we found that the expression of P66 downregulated with age. And we will know cardiomyocyte, the proliferation of cardiomyocyte is downregulated after postnatal. So maybe PC6 is related to the exact of the cardiomyocyte proliferation window, but is not confirmed. So we conduct our in vivo and in vitro experiment to confirm the effect of P66 on cardiomyocyte proliferation.
We have one more question. I see Dr. Chao Jun raised the hand. I will try to let her ask the questions. Let me try.
Sorry, I didn't. I just want to add another point to Dr. Huang. Another reason for she look at the Wnt signaling maybe because the Wnt signaling is already be found to be related to cell proliferation. So I just want to add this point. I'm also with Dr. Huang in the lab. Thank you.
Okay, thank you very much. You get one more question.
Oh, when not count the ratio of phosphorylated eggs and what's that? And thank you. And we have counted the ratio of phosphorylated ERK from our experiment. The data showed that PC6 downregulate knockdown have no significantly effect on ERKs or phosphorylated ERKs. And what is the significance of thank you. We have conducted the phosphorylated ERK and the total ERK in one experiment. But for showing, for our picture, show that ERK is on this picture and P and the phosphorylated ERK is on this picture, but the two picture is conducted in one experiment, and for first so we didn't separated counting phosphorylated ERK and total ERKs.
Okay, I think we will close the webinar today. And thank you very much for Dr. Huang for your nice talk and also for all the answers. And also thank you very much for Professor Nie Yu to let his PhD student to do the talk. Finally, I also want to thank for Jenny, the managing editor of JMCC, helping organize the webinar. And thanks everyone for joining. Bye-bye.
Okay,
Thank you,
thanks.
Professor Chen.
Bye-bye.
Professor Chen. Professor Xiao,
Bye.
thank you. And thank
Bye.
you for Jenny. Thank you. Bye bye.
Bye bye.